Okay, let's get it started. So this is week 11. Remember, we have 2 more weeks for lectures and also computation labs, of course. Then some of you start to worry about what presentation is supposed to be. So I released this in the week 14. So you will have a 10 minutes presentation here. So this is instruction. Any topics you want to talk. And so the normal flow chart will be background introduction of your project or your proposal, what you want to study using single cell data. Then you can discuss the significance of your problem you picked. That's very important. This cancer killed a lot of people, something like that. Then immediately it goes to the methods you want to use. And then you use PCA, you use Disney, whatever. Then your experiment design, then you present some of the data you collected. It could be you download and reanalyze. Then you use this different approach we talked about during the lectures. Then you see, okay, I get some results which looks different from a published paper, and why is it different? I found something new which the original author from the paper I downloaded data from, they didn't realize you can do it in this way, and I found very interesting. There's a new cell type they didn't pay attention to, something like that. Then you quickly draw a conclusion. So the single cell data analysis help you to achieve this. Or if you don't have a result, you say, okay, I expect if I have a data looks like this myself, and I can achieve this is using DV, study the program difference between genes. So that's the framework, the structure of the talk. You can be more creative. Okay, so for evaluation, so I will be here, but I'm not going to judge your talk. So you will judge between yourself. So everybody will judge another speakers, including yourself, and rank. So you believe who's the best in their significant peak of their talk, put rank 1, who's the second. So you have a 1 to 9 judge to others and include yourself. You can put yourself always top, but you average out by others. All right? So I will collect the ranking and take average. And for significance, for example, and then innovation, you believe you studied innovation, you put yourself as a rank 1, but others maybe put you 5 or 7. I will average them out and give you the score of your innovation, your approach, and your performance. You perform really well. Everybody believe you are either top 1, top 2. So your average will be really high in that category. Okay, I will be just monitor and modulator to ask questions, to suggest you ask questions. So performance including this 5 minutes questions between you and between members, okay? So that's the presentation. I need questions for this. Okay? If we have enough time, then just one day. And if we have 2 hours, 9 of you can finish on time. So you don't have to come on Thursday. Yeah, depends. If you were too slow, and then it should be okay. Maybe, yeah. Without a break, we should be able to finish one day. Yeah.
Which order do we present?
Will be alphabetic order of your surname. So I will give you a list. Yeah. I also released assignment number 4, okay? In here, homework number 4. So I ask you to read a paper. So this paper, then you realize, so normally single-cell data study is to find the samples, healthy disease, then annotate the cell type, then compare the same cell type between disease and healthy, okay, to find the difference. That's the logic. And you know how to do it, and all the technique is there. What I want you to learn from this paper is they do something from different angles. So they do this and then see, okay, we found the most different between disease and the healthy. I can find it from this particular cell type. So they flip around their question. See this, they define the question is not to find the difference between disease and healthy, in which cell type they found. They try to say, okay, if I have a disease, which cell type I should compare? I can get the most of the difference between the healthy and the disease. So they just change the angle to define their question. Then they create this cell type prioritization algorithm or pipeline. So I want you to read this to get some idea, say, okay, I just need to change my view of the problem and design a new study or new way of the thinking. So you have plenty of time to think about, give me an idea of a research idea. So it's the homework. Any questions for this part? Okay, so email me if you have questions about the last homework and also presentation. So today we are going to cover several topics in single-cell data analysis, including batch effects correction, okay, pseudo-time data trajectory analysis, and then we also mentioned this called immuno reservoir repertoire analysis. Basically, you have information you can get not only the individual cells, gene expression profiles, I mean the T cell, B cell mostly. You can also have their surface protein receptors, okay, T cell receptor, B cell receptor, subtype of the receptor colony. You can make the link and then see what you can get. So this is a very powerful approach I want you to know. So we will start with this batch effects correction, then talk about the CCA, then trajectory analysis or pseudo-time analysis, and then that's a repertoire profiling of immunocells. Okay, batch effects corrections has been a problem for a long time, and mostly start from the bulk RNA-seq. People think, okay, we have experiment have been done in one day, and all sequence on the different machine you get slightly different bias, the offsite of this profile overall. So you want to make a correction, make the same sample or same condition sample comparable to each other so they can compare with another conditions. So people borrow that idea and bike into single-cell data analysis. So you see this batch effect correction all the time from papers. So you want to have controlled and also treated cells and then try to align them. So make the overall profile comparable to each other, not just by the batch effects. So I believe all of you have already heard some of this, and the people believe if you don't have the data corrected, so you have batch effects in there, the difference you found will be biased by these batch effects. So people found the different algorithms in Surat. Combat is one algorithm has been used in the bulk RNA-seq. So it's directly borrow this, apply to single cell data. It's also work. It's working, and you just treat all the individual cells as a sample. So it was designed for before 3 samples against 4 samples, but now you just expanded this to several thousand samples to several thousand samples between individual cell populations. You can do this. And mutual near neighbors, we will discuss this. It's also designed for single cell data itself. So Dana Pierre is not a no person. So she's a senior research leader in she's in the Memorial Sloan Kettering Cancer Center. So she's a senior member, so developed a lot of the systems biology computational programs for single cell data analysis. This is one of the tweets she published a couple years ago. So batch correction is the worst for cancer because each cancer is different and unique. So what you see from the 2 samples, even from the same people in a different location of this cancer at a different stage, you always see some of this shift of the population of cell, cancer cell or associate cell types. But if you apply batch correction to force them to be aligned, you actually eliminate the most important signals between cells. So the purpose for individual cell profile examined and also using this single cell data, we try to look at the variability among cell populations. So if you force them, you actually eliminate a lot of the potential discoveries. But the reviewer always asks, you didn't do the batch correction. And I mean, it's really debating. So I never see the values for batch effects correction. So I encourage you, don't use it. Especially if you think DV genes, for example, last time we discussed, variability is more important than dynamic. If you force them to align and average out, the people also use bulk, pseudo bulk, so try to average 50 cells in the same condition and make this average. This is driven by the traditional way of thinking. So we have to correct this variability to make average. Everything has to be aligned before we compare. This is not correct. Okay, I still don't understand some of these batch effects. What's the purpose for this? People use a method called harmony, has been cited for thousands of times. We will run harmony, by the way, in the computer lab. I will show you this harmony will not change anything. It will not change the expression values. It's only change the embedding, the visualization, make a cell looks aligned together. Nothing else. I still don't understand the purpose of this, but people just use it. I believe their argument is for cell type annotation, but we can annotate cell if even they're not align to each other, you can still have, this is the same cell type, you merge them later. So anyways, but as a topic, I have to cover this in this lecture, because if you take the single cell data analysis, people ask you, I never told you about the batch effects correction. Well, I don't want to tell you. This is useless. Unless you believe that the technology is really bad, you sequence the same cell type in the Petri dish, for example, cell line. Today and tomorrow you get a completely different result. But that's not true. We tried. We sequence the same pipe cell line, and in different batches, there's no technique batch effects, because the machines are so consistent, even across different chemical versions. You have a very slightly the unaligned population, but for the downstream, really not very important. But people just develop those and consider using their mean-centered mind, so they only consider the average cross conditions. Nobody cares the variability is important for the function. That's why you have seen this all the time. So combat, as I said, this method and have been used not for single cell data, but has long history, but people just borrow that, directly use. This is one of the methods developed in right after the single cell data analysis become popular. And they very quickly put together some ideas. So this idea is very simple. You just have a different batch, so 3 cell type in different colors, and 3 cell type in different colors, batch 1, batch 2. From there, you found the nearest neighbor. So nearest neighbor is through the distance computation. So we know how to compute the distance. Euclidean distance, cosine distance, to have vectors. And so they just compute every individual cells with every individual cells cross conditions, cross batches, okay, not conditions. So that found, okay, for this cell, the nearest neighbor is this. So they make a line, make a link, okay? So that's one direction. They also compute start from this batch back to there. If these 2 cells found their mutual nearest neighbor, then make a link. So this is called mutual nearest, because from here you found the nearest neighbor is this. From here you found nearest neighbor is that. Well, this cell is not a mutual nearest neighbor, because it's only one way it found the nearest neighbor is this, but not from here. The nearest neighbor is not this pink one. It's this. Okay, so you just do this filter, and then you found mutual nearest neighbor pairs, then merge them. Okay, so after you push them together, basically it changes coordinates. Then you get this aligned, and you believe, okay, the batch effects have been corrected, because although they are away from each other due to the batch, but you force them to find the most similar cells and put them together back. Then if you have batch 3, then you do this again. So this new coming cell population will be merged or aligned into this already merged algorithm. So it's purely based on the distance. Okay, in lecture number 3, you learned how to compute the distance. As long as you know how to compute the distance, it's just do this cross the matrix of gene expression to found aligned cells. That's very easy. Another technique people use directly is called canonical correlation analysis, or CCA for batch correction. So we will introduce this as an idea for multi-domain data alignment as well. So it's not only for single cell data. So the idea is this: you have two samples, the treated and untreated. Then go through the pipeline, you get a matrix, and you found some kind of batch effects, you believe. But I believe this is a true difference between samples. But they call this unaligned data sets. So you want to have these two matrices in to give the CCA. CCA will help you to project their position and make them aligned. Okay, looks like this. This was separated before, but now they merged and then try to be together. Again, I cannot answer you if you ask why you do this. I don't know why they do this. Can you answer me? Anyone? I don't know why you align them. But anyway, they do this all the time. And then they align this and assume after aligned, and you can do a better cell type annotation maybe. Okay. Well, if you even don't match, you can still do this as the method I developed. This 50 cells, and you annotate. If this is a fibroblast, this is also fibroblast. You just merge the cell type together. Why you need to force them to be aligned first, then annotate the cell type? I don't know.
You just want the nice figures.
Nice figures looking, yeah. But that's not science. That's cheating. Yeah. Anyway, so another paper using CCA, they published in the same journal, and back to back with these two groups. But we are going to just introduce CCA as a method, as algorithm, just like we introduced PCA. So CCA, PCA are related, okay? So in PCA, we know we have this data, okay? We try to rotate and see, okay, this really looks really long in this axis angle, and then you can reduce dimension, put all the data project onto this particular PC. CCA deal with two data sets together, okay? So you can collect the data from one approach, okay? They could be completely different domain, and the data looks collected from different methods or different technology, completely different signals, but as long as you can put them into a matrix or vector, each data point is a vector, right? Then we can project using CCA into a shared common space. So you can do, for example, you measure something with 3 variables, but here you may use several thousand of variables, but they have 2 different dimensionality. But you can reduce the dimension both into 2, and using PCA, for example. Then you try to align them. So this extreme example is you have a picture, okay, like a photo of this lady, and you have soundtrack. They are very, very different data, okay, data type. But you know you can have a PCA, you can vectorize your pictures, okay? You can do convolution, you can do vectorization to make a PCA, this picture into a vector, the data, okay? You can also change your soundtrack voice, these figures, into a vector. Any data you can have. So you have a picture as view and the description of this picture. So you can tokenize this and make this as a vector and this as a vector description. So, and also this, the brain signals collected from EEG, you can use a functional MRI to collect data. They're same objects, different measurements collect different data, but those data can be vector. Data is vectors, okay, right? Everything is just a vector. You can change into vector. So once you have a vector, you want to have a first function to change your vector, to rotate, to project. Another function, so another function G, okay, do the rotation, transformation. Then you try to align them. So that's the idea of a CCA. So how CCA work? You can think as a two PCA. So you have data from one type, okay? The data from another type. Data points are vectors. So you do PCA on this, do PCA on that. But the next step is not only to show the most variable patterns in the data, but it requires your projection has to be correlated. So you project your first sample or data, X, into a long axis. You project Y into a long axis, but you have to have a regression. So this reduced X and Y should be highly correlated. So you want to achieve a high correlation between two data sets instead of PCA is doing for one data set itself, does not care the another. But in this case, you want to rotate data one, okay, one data align with data two, so you cannot using single sample, single data PCA to do this. You have to consider both rotation at the same time. You want to achieve the largest correlation after they project onto reduced dimension. Try to make this regression as large as possible. You try to maximize this formula. Okay, this is a correlation coefficient. But the idea is the same, and as a PCA, right? You know the PCA is a rotation, or try to find the rotation matrix. But here we need to find the two rotation matrices, one for each data set. But our goal is to make their projection alignable. Okay? So how to achieve this? We needed to have this mathematical expression to try to maximize this correlation after X and Y has been rotated with A and B. So there's two rotation matrices you can consider. So A and B has to be chosen in order to make this correlation maximized. All right, in PCA, you don't have to care Y matrix because you only work on the one single X matrix in order to rotate it to show the largest variability. But now you need to consider two together to get the maximized correlation. So that's the idea. So how to compute the correlation? You do this after rotation, then this is a correlation coefficient. Involved this covariance matrix and the covariance matrix. By the way, you need to centralize the data, okay? So x has to minus the average, x, y have to minus y average. So this centralized data correlation, okay? So this is the algorithm. So if you give this algorithm to AI, AI will help you to code, right? You know that you can do this. I encourage you to use because everything here become transparent to language models now. And you can use language model to help you to translate into any language you're familiar with. I believe a lot of teacher don't want to use AI because there's a maybe replace your thinking, but I believe this can help you to think to some extent which you'll only be able to achieve without AI, okay? As I said, if you give this to AI, it will help you to implement, and you will see, okay, the code make more sense compared to those. But you have learned SVD, you have learned covariance matrix, you have learned the centralization. So everything put together, not too hard. It's all algebraic operations, okay? At the end, there's matrix multiplication. It's at the end. So that's a MATLAB function, canonical correlation. X and Y, and then you have A and B. I believe A and B are so those are the rotation, the projected. So A and B should be aligned version of X and Y. So, okay, it should be alignable. So in order to align them, so this has to be, I believe X and Y should be the same samples, same number of samples. We can try and see whether X and Y should not have to be same variable. So you can have a 3D definition for X, 4D definition for Y, but I believe the sample size should be one-to-one aligned. We can see. Okay, so as I said, PCA and some others as well. So this is called a multivariant linear regression, MLR, and the partial least squares. So they all linked somehow. Okay. So when we talk about PCA, we care only one sample. So A, we don't care B. So you only look at the covariance matrix for the variables in A, but they put an identity matrix for B. So if you put all these algorithms into the same umbrella framework, they involved A and B, two samples, and the CCA will be something like that, okay? And put together A and B. And the PCA, because we only consider one sample, so B will be the identity matrix. Well, PLS, you will deal with something like that, A and B, and multivariate linear regression will be something like that. So these two matrices put together and under the same algorithm and reduce into the single eigenvalue decomposition problem at the end. It's just set up differently, and you get the solution same for those algorithms. Okay, so CCA is algebraical operation. So just like PCA, just rotate and found two rotation matrices. And you can also, if you don't want to, if you, for example, you have no idea about those rotation matrix and everything, you can see, okay, my goal for CCA is just try to find the best parameter in order to align these data points from A and B. So your problem become a optimization problem. So if you can describe this cost function and you don't have to go through those formulas, okay, you just say, okay, I know I try to make this alignable as small as possible, make this distance across all the samples as small as possible. You can get this A and B by put it into an optimization framework. CCA also have this conventional way to do this, and sometimes you want to do this transformation, okay, to make kernel version, so kernelized to, if you cannot do the alignment because some this nonlinear components in there, so you just no way you can align them, but you kernelize, transform this low dimension to higher, and over there you can get the CCA better. There are also some other variations. It's called sparse CCA or probabilistic CCA. So those are the ideas. So this is a kernel version of CCA. So this is a non-kernel version, just regular, and you try to maximize this correlation. And here is kernel function. You basically you do this kernel transformation first, then put that into. There's another CCA. It's called a canonical correspondence analysis. So you need to be very clear. This is very different from a CCA. Okay, so somehow the people in the ecology study use this technique. Just some terminology, so don't get confused whether it's canonical correlation analysis. That's what we talked about. It's related similar to PCA, but applied to two samples to align them. But the correspondence analysis is a very different technique. Okay. Another terminology is called dynamic time warping, DTW. So this is a method to align time series data. Okay, so you have this and that, they are in a different lengths. It could be collected at a different frequency of the same signal. So that's, for example, you have X, it's a vector you sampled along the time. And this blue curve is X. And then you have Y. But it's sampled shorter and or more frequent. But you get this different how to say layout or plotting out. Then you apply a DTW algorithm to these two vector, to time series data. And this will help you to align one of them onto another, okay? So that's the result. And you found the minimized distance and this new X and Y and along the time. So how this works? It's combine this distance matrix counting. Okay, so you have this, that aligned, then you found the distance, the shortest path, and then in here you have a 3-step jump. That means 3 of these points in the time series 1 has to be aligned into one position in the time series 2. So you do this align and then move forward, keep aligning, and then you get the final alignment between 2 signals. Yeah, so those are the terminology or technique you can put back on your mind, and maybe some study you believe, okay, why we just align them first, then do something else. So when you need them, you have that knowledge. So like your homework, if you get a new idea, okay, I should combine this together, I can solve a unique problem that cannot be solved before. So you need a lot of those. You have a PCA, you have a CCA, you have a DTW, you have what else? Eigenvalue decomposition, you have many others, right? We discussed. So how you creatively or innovatively combine them into a new algorithm of help you to solve the problem from different angles. And also you have a language model, the AI, to help you to implement any idea you have. You say, okay, I put DTW before PCA. Oh, I do this. You don't know how to code, but as long as you have this idea, tell the AI and help you to build this pipeline for you. But you need to have this foundation piece and to thinking. Otherwise, you don't even have the keywords prompt to give to AI. That's a problem, right? Okay, so cell lineage tracing. So this is a technology people want to use to follow the development stage of the cell through maybe image process technology, or barcoding, or some dye labeling, and the people try to follow those in order to study, for example, embryo development, this blood cell, so the homeostasis tissue turnover, stem cell, so how the stem cell differentiate into matured cell types. So if you have a way can do this, it's a very powerful way. So in this case, for example, image-based genetic barcoding, okay, you have a special barcode and to put into the cell, and then you can follow, okay, with this barcode there, you know particular tissue was derived from that chimerical, the cell you introduce into their other systems. Case 9, okay, the gene editing based. So this is a genome-wide association study. In single-cell data, we cannot do it because we collect samples in one time point, although we have thousands of cells in that sample. But here, the idea is try to use a computational technique to align cells and make a pseudo trajectory, and then see, okay, from this end is early stage of development, and that end is later stage. So I can observe some intermediate stage. So that was very nature thinking. And once you have a lot of cells from the tissues, then you try to do this, okay? Try to rebuild the possible development trajectory. But because they are not real, so people say, okay, this is a pseudo time or pseudo temporal order. So that's the idea. So this is called the Waddington epigenetic landscape. Basically, you have a cell type, for example, then it can differentiate through different trajectory. And they're very robust. Basically, you just cannot go, no cell will go from, for example, this ridge here. They all go through this predefined valley and then become the cell type which will have its function. So if we have this on mind, we believe the cell will follow this trajectory. If you collect enough samples from enough cells, some cell will be in the intermediate stage, okay? It's not everywhere, just like something not controlled, but all this canalization is highly genetically controlled. Maybe epigenetics play a role here, so you will never have a cell which jump out from this canal, and you always go certain direction. So that's why this is the genetic background or biology believe that's the case, so we can infer the trajectory. It may have a branch, okay, goes that, and will be a very interesting topic to see, okay, what's the cell type or cell states looks like before they decide to branch out into this trajectory or that. People have done this. Of course, you can collect samples from a different development time. So you have samples from after fertilization, 24 hours, 48 hours. You can collect multiple time points in the real time, but for the sample you collect at the same time, one time point, you can still try to build this trajectory to recreate statistically inferred order of cells. Then follow this trajectory, we can look at genes expression, whether some genes going down along the time, some genes go up, and some genes maybe show nonlinear patterns and only go up at a certain intermediate stage of development and take effects and then shut down. So we are interested in these different patterns of the gene expression along the cell. So give you some power rather than just compare starting at the end. But now we introduce intermediate stage. With intermediate, we can have early intermediate or later intermediate. You can have a lot of pseudo time points. It's a very powerful way to do this. So that's early stage. People publish this. They collect stem cell development, and they found it goes to different directions. There are breaking points, and the cells follow. So that's very early stage studies. I think this study found there are different trajectory if you induce this using different factors. Okay, it can go this or that. Yeah, I show this picture. Just if you imagine your single cell data is a picture like this, you know you cannot. You need to take multiple pictures in order to combine them to make this final But the single cell data, just one sample, you will get this picture. So if you can work out pseudo time, and you can have that reconstructed of some dynamic process. That's another example I want to show people. Single cell data is a static single time snapshot. There's no video, or you can take how this horse is moving and its gait. But if you have a picture of this many horses together, you align them, probably you can reconstruct this movement, the gaits, how these legs move, just like you have a video recorder. So that's the idea for put the t-SNE, the static, into elongated something like this to reconstruct this trajectory. Okay, so people found a lot of algorithms try to move away from this static snapshot view of the cell, but try to build this elongated trajectory out of it. Okay, so they just use any algorithm they can find. So one of the algorithms is to in this embedding, okay, they use a PCA. They start to build networks. So you believe there's some center of the different stage, then you build a network, something like this. Then using your knowledge of biology, say, okay, this is the center of the development. So from here, you just move your step, follow this network, and you believe I should have a trajectory move from here to this, and come back to here, and then there. Then you build a trajectory. So network-based, okay? There's no good reason why you need to do that, but some of this biology knowledge may be involved there. Maybe a marker genes for stem cell expressed highly in the center of this embedding. Maybe here, okay, or in this case, it starts from one end. And you know some mature or fully developed cell type have markers at that end. So you have some idea of this possible trajectory along this cloud, okay? Anyway, there's no fixed way you can use to see, okay, this is real, but they just try to approximate this, estimate. And the algorithm will be help you to make this easier, okay? So there are many pipelines has been developed. There's no particular reason one pipeline is better than another. So one's maybe convenient than another, or more popular than another by historical reason. So those are some select ones, okay? So first, they all start with a dimensionality reduction. They all start from PCA, t-SNE, UMAP. And I suggest to use FATE. FATE gave you the better linearization of those population. And t-SNE, definitely we should not use it. t-SNE is random. You should put one population cell here, another here, or you put another here. It doesn't matter for t-SNE. You start from a random number and you put them as long as they're separated. So the link between subpopulation in t-SNE, same as the UMAP. I don't suggest you use UMAP to do this trajectory analysis, because the position of the population subclusters doesn't make any sense. This position is far away from another cluster. That doesn't mean this is more different from another one. Anyway, so they use different technology to do dimensionality reduction. For example, use a diffusion map or something. Then here they build a network. Here they use curve fitting or construct center of clusters. Then try to, for example, follow this cell cell link between cells to make a trajectory. And here there are some collapse individual cells population into clusters. And then here they use curves, trajectory curves. One of these show up the algorithm called the minimum span tree. Okay, so you have those different clusters. Then you try to build a tree, and the tree link them all. Okay, but the total length of this tree has to be as small as possible. So you want to build a tree link all the nodes, all the subclusters in your population of cells, but try to minimize this total length of the tree. So once you have that tree, and then you can start from one end to build a trajectory, and you can have another trajectory and different parts based on your understanding of the population of cells. Those ones are using nearest neighbor graph to build a network for cells. Okay, so they found nearest neighbor from one cell to another. If they're nearest neighbor, then you make a link, and then at the end you build a network. So this network, for example, will not link this cell with this cell, because all the nearest neighbor of their not shared, so they have to go through these parts and move from this cell to this end. So once you have this nearest neighbor network of cells, you can start to build, for example, using this minimum span tree put onto it, and follow the tree, and to find this trajectory. You can have one and have multiple. So this is an example of the minimum span tree nearest neighbor graph. I don't know if I can run this. It's Java. We run this and then take a break, okay? So those are the random generated 50. You can change this to 66 points. Then let's see, I will choose the nearest neighbor by 2. I link K. So KNN, you know, K is your one node found how many nearest neighbor. Okay, 3 nearest neighbor. They link 3 of them. So you make this K bigger than 4, and then you make a link between them. 5. Okay. If you put 1 here, so they only link one node will only find the nearest neighbor by, so these 2 are the nearest neighbor to each other, so they only link by 1 step or 1 neighbor. But if you see, okay, number 2, 1 node will find 2 nearest neighbor make link. So you use this way to make this KNN network. Then you move along this network to achieve this trajectory reconstruction. Okay, let's take some 5 minutes break, and then we continue. Okay, so this is another example of KNN graph. Based on the number of K you selected, if you only consider 3 nearest neighbor and you have this nodes which isolated from the rest of them, because you only consider 3 nearest neighbor, they all has been inside this small island within themselves. But when you change, say, allow 4 different nearest neighbors, then you make this KNN graph would see denser or more connected than this particular island will merge into this large KNN graph because you consider top 4 nearest neighbors. So we use KNN in this pseudo-time data analysis. And also KNN is important for our next week's topic. It's manifold alignment of those, because we want to learn this small transition between one stage to another. You through this KNN bridge and move from one stage to another by small steps. But after you accumulate enough of those small steps, at the end, what you get is very different from your beginning. It could be through this highly nonlinear structure, which you'll never be able to through the jump from one step to another stage to another. But if you follow this KNN network, you can merge or move from one location to another. This is an example I built from the real data. We will try this in my lab tomorrow, Thursday. So each position here is a cell. There are some outlier cells, but most of cells will be inside this cloud. And then you build a KNN network, and you can ignore, for example, those ones, and you build a trajectory, go through this network starting from here, and then you move along the network and then end up to here. So if you have this, you can see follow this trajectory, and the cells will be aligned or collapse into this trajectory. Then we can look at the genes, and some genes move from here, this end to that, will show monotonically increase of their expression. So this gene probably can tell you why those cells have been pushed into that particular corner in this KNN graph. And some other genes will go from up regulation to down regulation. So if you know those genes, you will see, okay, at the end of this trajectory, and the cell become highly expressed for this gene, lowly expressed in that gene because of this particular process of regulation. In this case, is NF-kappa B regulation. As I said, in the data science doesn't care what this idea. They just try to find a way which can represent your data. And in our usage here is to try to find the trajectory among your data points. So you can use any algorithms you believe can achieve the goal. So you do this linear reconstruction based on the cluster networks, based on the, this is the RNA velocity, which use splicing information, try to build, okay, which gene goes to which direction to project its directionality. I don't think I will spend too much time on this RNA velocity, given the time of maybe we discuss this Thursday. But let's jump to most commonly used pseudo-time analysis. People use monocle. the most, I believe. However, Monaco is very messy package or methods. It has a different version. So this is version 1. They use ICA to do dimensionality reduction, then use minimum span tree to build this network, and then move from the minimum span tree to get a trajectory, and then that's first version of Monaco, published 2014. So remember the single cell data, the device of 10x Genomics released 2016. So this method had been proposed before the real single cell data become available. Okay, they probably build on some hypothetical simulations or short small scale lab studies. So they come into the market very early, then released Monaco version 2, and here they used reverse graph embedding. So they abandoned the ICA algorithm, but use another, and then see, okay, this is more accurate, robust, but nobody knows. Then try, see, Monaco is better than Monaco 1. But this is a reverse graph embedding. I believe this machine learning technology to learn a parsimony principal graph. Principal graph is a principal curve that passes through middle of the data, allows some have a branch. Okay, so by default they will use this DDR tree and then scaled reverse graph embedding. So this only is an option for the authors. And then this model 2, and then go dimensionality reduction, and build this, and then principal trees. But this is some of the combinations of data science technologies. And then release Monaco 3. This is not formally published, but now algorithm under the hood changed again using clustering. I believe they try to make Monaco 3 a comprehensive package, not only to do the trajectory, but also clustering and also differentiate expression analysis. But I believe this is wrong direction, because if Monaco is famous for the trajectory analysis, you keep the similar function through the different version, or you call something else. You cannot use Monaco 1, 2, and 3, and a completely different implementation under the hood, and also try to become comprehensive package. And here the clustering using UMAP and low V community detection algorithm. But anyway, Monaco 3 is the one I incorporated into MATLAB. So we will play with this Monaco 3 Thursday. And then I will show you using the manual draw of the trajectory. We abandoned any algorithms. We just use fit. And I show you how to draw this trajectory by hand, okay? We use a mouse to try. Okay, so just very quick idea. So another, maybe after Monaco, the popularity, and then slingshot, another algorithm. They use principal curves and also minimum spin tree and to achieve this. So those are the summary. They believe slingshot will be able to work to any dimensionality reduction, and the cluster-based use minimum span tree on the clusters to get this graph, then principal curve. So that's a feature compared to other, for example, Monaco. Monaco 3, by the way, has not been published. So it's only changed in the package. And this is a snapshot, maybe more robust to noise. They show some examples. And they can detect these branches. So you will go 1 trajectory, 2, and 3. Slingshot will help you build multiple trajectories. Well, Monaco, I don't know, this is Monaco 2 or 1, so maybe 2. I can only build major ones. Another is called T-scan, pseudo-time trajectory construction algorithm. And this is a nucleic acid research paper, 2016. And the paper very detailedly, in detail to describe this algorithm. So it's very easy to re-implement. Stream, another one, okay, same flavor. Lisa, it's another one. I don't know if it's published yet. It's in the preceding documentation, but they are KNN based. This isomap, then pseudotime. So you can see everybody has its own idea on how to manipulate, move, and set up this. You have many, many different algorithms. So this is a review paper, try to summarize, see which one overall performance, accuracy, scalability, usability overall is better than another. So they try to put them together as a review of the different methods. Graph-based, tree-based, multi-factory, frication-based. So this is continue the same big table. Then they have a evaluation criteria for accuracy, scalability. Okay. The big table continues, and then there are different values, scores, and different colors to give you ideas. At the end, they give this practical guideline for which method you should use, whether you want to consider the branches. Some algorithm just not take care of this branch, and whether you believe it's graph or tree, and so there are different options. And there's a package called Danvers. Okay, so you try to, it's our package, try to put multiple implementation under the same umbrella of it in this package. So internally, they install a lot of different algorithms together. So you install one, and you can try different algorithms. So once you have this pseudo-time assigned to cells, then a lot of downstream analysis become available. So if you don't have the reconstructed trajectory, you don't have this pseudo-time time theory information. So a lot of analysis you cannot do. But now you can follow this pseudo-time trajectory to compare, for example, starting point to end. You can compare to the intermediate stage. So differential expression analysis will have multiple time points to to do, and you can have a visualization to find those change of gene expression changes along the as a function of time topology of cell state transition. Okay. Again, that comes in the biology, single cell biology. Let's say pseudo time is not a real time, okay, not an actual time. A lot of this inferred results, downstream analysis, may lead you to wrong claim or pseudo claims, or just keep that in mind, and they're not real, and it's helpful, but maybe not real. So one of the idea here is, so I draw this myself, but never implement it into anything. So I want to follow this trajectory. So you build this trajectory, then I do sample using a sliding window. See, enough cells will be sampled in time window 1, 2, and follow this trajectory, sample a lot. Then for each population of samples, we can build a gene regulatory network for each. So now you have a time theory of network. And this network become a 3D structure. So if you consider network as a matrix, now you have a 1 matrix, 2 matrix, 3, and then it become a 3D tensor. And this tensor can do decomposition, just like you have eigenvalue decomposition. This tensor can be decomposed into different components. Then you can do, see, which components are follow the time, which are not moved in what kind of patterns. Okay. So the example for to show you can study specific time cell type. So if you extract neurons from this data, you can do this pseudo time, and you can build a network. And this could be different layers of the neurons in the central nervous system, brain. So I told you we won't have time to do this RNA velocity, but I will cover that in Thursday, on Thursday maybe. So this involves splicing. So you have 2 matrices count for the spliced, unspliced, 2 different matrices each sample. Not like a single cell, it's just only one expression matrix. This expression matrix will separate into 2 matrices in order to do this kind of analysis. So I'm going to move in the last 30 minutes to talk about this immunorepertoire analysis receptor. So immunocells, we talk about this T cell, B cell. They all express receptors on their surface. So those receptors show the high diversity of the subtypes. Okay, those receptors have its own different arms and they create different colony of the B cell. So B cell itself may look similar, especially if you do RNA-seq, single-cell RNA-seq for the nuclear genes. But their surface receptor will differ from one cell to another a lot, okay? From this generation of this. The T cells are the same. So you have T cells and different surface receptor protein. And they create a different affinity and to talk, to combine with a dendritic cell, the presenting the signals once they have interaction with pathogens or other foreign objects in your immunosystems. environment. So this communication will trigger a signal, say, okay, this is very useful to fight, for example, this particular pathogen. And this signal will make T cell proliferation and expand its colony of this particular type. Okay, the T cell receptor will have different subtypes. If there's one and the fit combine this dendritic cell representing signal strong enough, it will stimulate the growth of this particular type of T cell, expand its colony. So the diversity is from the T cell receptor, because this receptor contains these different arms and long and short. That's also there's variation fragments. So through this rearrangement, that you create different subtypes of the receptors. Okay. So these subunits are defined in alpha, beta, gamma, delta chain, different variations. They are similar. They're same gene and parallels, but define slightly different sequence. So the combinations of those will create a highly diverse population of the T cell receptor differences. So T cell receptor contains mostly will be alpha beta, so alpha beta subunit. There are also delta gamma, so delta gamma is two different arms in there. Well, alpha beta gene, for example, for the receptor subunit, alpha there have V segmentation domain. There are different, 70 different ones. For the J subdomain segment, there are 61. Then combined with beta unit, their V domain segment have a 6-7 different types, and their D segments 2 and 13. So they can combine, and using one of each, okay, to combine into different type of alpha and different type of beta domain. And this domain will create, so this is a place where this alpha, I believe that's TRB, so there's a beta domain variation in encoded gene in the chromosome 7, that's human data. You see these different subfragments. So T cell receptor for particular T cell will select through this recombination one of them, and will be combined with other segments and together. So you see the beta domain and on the 7 chromosome 7, and also alpha domain was is in the chromosome 14. Okay, so those combinations together. So this is a place where 14, that's alpha domain. So you see all these receptor subunit domains. Okay, there are many of them. This is in chromosome 7, and that's gamma domain. So 7. If you come back here, the gamma is on the 7. They're all there. Okay. So those subdomains fragments will be selected through the recombinations, okay, assortment, rearrangement will combine and create one of the unique combinations for each T cell beta gene. So this is only one arms. So you have this 10 to the 14 combinations, and because the subdomain combinations together, you get 10 to the 21 pairs of possibilities. So this diversity is large enough to help them to have different levels of affinity with different type of this pathogen presenting signals. So we want to sequence not a T cell itself, but also sequence which of these particular combinations of these subdomains in their receptor. So that's why 10x genomics have this key to specifically work for T cell receptor gene amplification or B cell receptor gene amplification. So if you want to do this, you have to buy 2 kits for your, if you want to study, for example, T cell. You want to sequence the T cell using the single cell RNA-seq, the one we just talked about before. And then also one of these T cell receptor amplification kit. You do that twice. If you want to study B cell, you will buy another B cell receptor. So if you have samples, I have a immunocell samples that contain both B cell, T cell. Then I have to buy single cell RNA-seq kit for this population. And then I want to have 2 T cell receptors. I have to buy T cell receptor kits. And I want to study B cell, then B cell receptor kits. So now you have 3 kits. One is single cell RNA-seq, another B cell receptor, another T cell receptor. Okay? The cost will be 3 times. Okay? Single cell RNA-seq. That's one kit. You get the B cell, T cell. Gene expression by this one kit. Then you have the receptor, B cell receptor, T cell receptor. So you have 1 kit, 2 kit, 3 kit. So very expensive because same population of cells, you have to do 3 times. Well, so this study, why it's important? It will give you the surface protein for this particular T cell. What type, if this belongs to alpha beta type of T cell, which V and which J, which VD, which J, which segment is there exactly. So you have that information. So how this 10x genomic kit achieve that? So this is a single cell RNA-seq. So this is that part, okay? So we see this before, so that's in the lecture number 1. We will label them, use barcode and UMI. Barcode tell you which cells come from, UMI tell you which gene is the count of genes, and those poly A, poly D tails tell you the gene fragments. They're all the same so far. They all captured using the droplet, generate barcode, and do this all the same. Now the difference is, after this step, your barcoded cDNA already prepared. Okay. Now you are going to separate them into one for gene expression, another for either B cell or T cell receptor. You separate your prepared. By the way, it's already, let's see whether it's already lysed already. So this cDNA. Yeah, so the cell should be already break up using enzyme. So everything should be released already. And let's see, try to figure out whether this pool has been pulled together.
Do we have to sort specifically for the T cells and B cells?
Yeah.
To sort this for the cells.
Yeah, so you have to sort. Yeah, I believe at this stage, all the droplet already mixed because the droplet would capture the cells. Cell have been released and mRNA in the droplet. But now also the droplet, everything have been put together already. So this soap contains mRNA amplification for all the genes, including these receptor genes, B cell receptor genes, everything already in the same soup. So this soup will be separated into two parts. One will go to amplification of T cell receptor, another round. Okay? So this soup contains all the cells, all the mRNAs labeled, all the cells together. Everything is one pooled sample, cDNA. Then this soup will be separated into two. And this can be sent to sequencing to get what we single cell RNA-seq matrix, data matrix. But this one will go another round of amplification for T cell or B cell receptors genes. You will also get T cell receptor gene expression here. It's already have them here, but not detailed enough, not to have a rates which can tell you how the receptor gene subdomain combined. So this only gave you the count. See, this have T cell receptor, B cell receptor gene, but only single numbers expressed at a certain level. But this part will go through another round of the full T cell receptor gene sequencing, basically, amplification sequencing, to get the full length of that particular receptor gene, so that they can tell you. For example, this is this part, okay? And the user kit, you have to buy this to amplify, to do this part, amplify this part, to give you VDJ, the rates. So this another round of application will give you the information about the VDJ segments information from this enriched T cell receptor, okay, or B cell immunoglobulin transcription. You already had that information there, but here one more step, amplification, to give you the full length rates of this gene, not only expression level, but also which type. Now you put them together. So this is single cell RNA, the gene expression. The bottom one is this only. The top one which through another amplification, so you get information of their receptor subdomain information. Okay, so Cell Ranger have another specific version that for take care of those. It will start to count and also assemble the fragments information to categorize them into a different group. So you can see this called contig. So this is one of the T cell or B cell. It's T cell here. Then they have this particular contig, which long enough to tell you this is it's alpha chain or beta chain, which V gene. So this is V number 3, and the J is number 27. And another T cell may have this V number A is V9, and then 49. So every T cell, every B cell will have that information. Okay, so this table, this annotation is what you get from using this extra kit. You don't have that table if you only use this, because that T cell receptor is only one single number expression level for that particular cell. But now you have this particular cell, but you know this. And in the file, you have this full length of each receptor alpha, beta chain. So you have those. And you can recognize this is from, for example, V5, this from D7, and then the VDJ. And the combinations you can read out from the. Why is useful? Okay, so now you have your T cell population, and you also need to know their clonal type by looking at their combinations of those fragments. And then you recognize all these blue ones share the same subdomain. So that means the population you sampled from the T cell for that particular sample, they dominant by only one subtype of this colony. If you know this sample is challenged by, for example, HIV, and you know, okay, T cell is fighting using these particular combinations of T cell receptor to deal with this HIV infection. If you have no infection, you probably have a very highly diverse baseline of this diversity. But until you have the challenge of the pathogen, and one of the colony will be selected, amplified maybe, several magnitude of high in the frequency. That's what you observed from this particular study, which majority of the cell, T cell is one of the colon type. And then if you have enough information, you may figure out why these particular combinations of subdomains response to that pathogen. If you can repeat in a different batch of experiment, you always see these particular combinations of the T cell receptor or subcolony of this T cell get amplified by the system. Once they see this pathogen, they always have that. That's a very strong indication that this T cell receptor is, the pathogen is a determinant for amplify this particular T cell receptor. Okay, so this picture is the same as this. It just show you the different colony clonal type and the different numbers of T cells. So this looks like, to me, it's unchallenged. So you have a different subtypes of this. But if you have only one subtype, then you will see all of them will be merged into just one major one subcolony. Okay, that's all for the lecture, and let me know if you have any questions. This is very powerful analysis, but I haven't seen a lot of discoveries, and also nobody in use of this on our campus, as far as I know. Okay, no questions, good. No?